Well, welcome everyone. Welcome to the April webinar. Thank you to ISEE for co-hosting this webinar with ISES. My name is Matt Strickland. I'm professor of epidemiology at the University of Nevada, Reno School of Public Health. I'll be today's moderator. We have two presentations and then we'll have some time for Q&A at the end. We're going to hear a presentation from Dr. Anna Rappold. Dr. Rappold is statistician with the clinical research branch at US EPA. Anna conducts clinical and epidemiologic research on health effects from air pollution and other environmental exposures, and has authored several studies specific to wildfire smoke impacts on cardiopulmonary health. Anna received the 2019 Arthur S. Fleming Award for the Smoke Sense Project, a smartphone citizen science app that seeks to answer how people experience wildfire smoke and builds community around health protective behaviors in the face of exposures. We will also hear from Dr. Luke Montrose. Dr. Montrose is an assistant professor in the Department of Environmental and Radiological Health Science at Colorado State University. The Montrose Translational Health Lab looks at environmental toxicology questions through a broad lens from cell to society. Luke's research focuses on understanding the influence of environmental exposures, such as wildfire smoke, on life course disease risk. In the lab, he utilizes tools such as epigenetics to characterize exposure impacts, and in the community, he leverages low-cost air monitors to measure exposure risk among vulnerable populations. So quick, some housekeeping things before we get started. Throughout the presentations, you're welcome to type a question either in the chat section or in the Q&A function within Zoom here. We'll review those at the end. If you prefer to ask your question in real time, we'll have some time for live Q&A at the end. Our preference is to have both Dr. Rappold and Dr. Montrose do their presentations, and then hopefully we'll have a good chunk of time at the end to do questions. So that's what we're doing today. Anna, why don't you go ahead and share your screen, and whenever you're ready, go ahead and start.
All right. Well, thank you, Matt, and Sarah and Nicole for organizing this webinar series, and for the topic, for attention to this topic. Can you hear me well? Just to check before I start talking.
Yes.
Okay, good. Okay.
Anna, are you there? Anna? We can't hear you.
I'll send her a message as well. Oh. Anna, are you there?
All right. Well, let me start by kind of looking at the air quality trends since the implementation of Clean Air Act. Many of you probably in the audience have contributed to this figure and to the steady improvement in ambient air quality over the years. It's certainly true that there are areas in the country where work remains, but overall, on average, air pollution across the United States has been decreasing, or equivalently, air quality has improved. Now, meanwhile, due to increased incidence of very large fires, many parts of the country, particularly West and California, the air pollution from smoke has reached new heights. In the US, wildfire smoke is now a single largest source of particle pollution, contributing to anywhere between 30 and 44% of particles in ambient concentration in recent years. So naturally, we invest a lot of time studying the risks and the factors that contribute to this vulnerability. We know a lot about health effects of exposure to ambient air pollution, right? From our decades of studies from urban environments. But a few decades ago, when these trends started, we didn't have very many studies of wildfire smoke exposure and their impacts. So let me use first a few examples from recent years on how knowledge gaps has been slowly filling in. One of the first knowledge gaps that was observed was that we know cardiovascular effects have been clearly linked with, causally linked with air pollution exposure, right? So, but a decade or two ago, there was not very many studies about cardiovascular effects. So there was a question arising, what would be, why that is so. So we invested, some time, several years of studying cardiovascular outcomes. And they're also very severe. There's severe consequences of cardiovascular outcomes, and they're prevalent, so it makes sense to study them. So in this study, we examined all emergency department visits in Northern California during 2015 California season, wildfire season. At the time, that was the largest smoke season ever observed in California. But since then, it's been surpassed several times over. In this study, we observed strong associations with respiratory outcomes at all levels of smoke exposure. And even for younger adults when the smoke was denser. That was not surprising, right? But what we also observed was, looking at cardiovascular and cerebrovascular outcomes, the strong associations with smoke exposure among older individuals. It's just one study, but this study did clearly show that the risk is differentiated by age, and that cardiovascular effects were observable. There was also a gap in the knowledge about mortality. We knew a lot about urban air pollution, ambient air pollution, and the effects on mortality, but not in the context of wildfire smoke. That's probably still largely a gap. A few years ago, we looked at one of the most vulnerable groups of individuals, that is, individuals with chronic kidney disease, specifically those who have their chronic kidney disease managed with hemodialysis. There was, we identified nearly 50,000 deaths over a period of 5 years and across the country from US renal disease registry. So it was a complete count of all deaths in this patient group. And we reported 4% increase in daily mortality per 10 micrograms of increase in wildfire PM2.5, which was model estimated, and 7% increase in mortality over 30 days after the exposure. On the days when there was significant wildfire smoke exposure, up to 8.4% of daily mortality was attributed to PM from wildfire smoke. And this study highlighted an immediate and persistent effects of wildfire smoke, and also identified this group of patients as very vulnerable, right? They're at higher risk than than healthy population. So over time, as the evidence from research emerged, the research grew as well, and our capacity to study various outcomes increased. So to detangle the impacts of of wildfire smoke and PM on various outcomes, we then became able to study outcomes that are less prevalent. I mean, they're more prevalent, but less severe than hospitalizations and mortality. Last year, Stephanie Cleland published a neat study of cognitive performance in the working age population and exposure to fine particulate matter and wildfire smoke. This study was published in EHB, and she used data from Lumosity, which is a smartphone-based application, a game focused on kind of like oriented toward the attention, helping people build skills to like improve their attention in working age adults in longitudinal setting. And a neat thing about this study was that we had exact time step of when each test was done, or each input to the game was done. So Stephanie was able to show that the strongest associations were observed within very short windows of the exposure on the hourly basis, and the strongest PM associations were actually more pronounced in the areas impacted by wildfire smoke. She also then observed the strongest association in younger adults as well as older adults. Now, I use these three examples because we can go back and review evidence from recent studies for days, right? But I use these three examples because you can start seeing a big picture that emerges. Well, there's many factors that increase the risk of adverse health outcome when you're exposed. These factors are not unique to a small group of individuals, and there's not specific outcome that's associated with PM 2.5. These factors are shared across populations, and almost no one in the end can completely escape the impacts during the wildfires smoke exposure. Now, while there is no simple solutions or quick solutions, practitioners and researchers and policymakers have been proposing a number of paths forward. In the area of exposure reduction, through public health messaging, that's been very active area of research and activity. They focused on providing reliable information and access to resources that would allow us to make timely actions, learning how to incorporate living with smoke into our daily activities, develop new habits in a health protecting manner, and then ensuring that activities and recommendations that we make are consistent and cohesive at multiple levels of our society: individual, interpersonal, organizational, community, policy levels. All that has to kind of be coherent, right? On the risk reduction, there's also been a number of methods proposed, including prescribed fire, mechanical clearing, and all kinds of other aspects. So from how this has been evolving, one thing that has been clear to us is that knowledge gap exists on providing the exact information on how successful different resources are in actually reducing our exposure and improving public health outcomes. So this is the context of the study here. In this study, I will focus on understanding the trade-off between increased use of prescribed fire to reduce the risk of wildfires and of catastrophic wildfires, that is, and the added health burden due to additional increase in smoke exposure in the state of California. In the state of California, recognizing this threat, the state is planning to dramatically increase the use of prescribed burning to up to as much as 1 million of acres annually by 2025. Okay. So this project is kind of multifaceted. It has different levels, but including smoke exposure modeling and community engagement. But here I'm just going to start talking about the health analysis, which tries to understand what portion of the health burden can be attributed to wildfire smoke and to prescribed burning? Is the smoke attributed health burden distributed equally across demographic and geographic strata? And then will the future exposure that we are intentionally bringing on, is it differentially impacting any specific groups, right? Because there's an opportunity for us to build in measures of resilience there. And having said that, the study overall focuses on, does this health burden analysis in the historic period and in the future period. But here right now, I'm just going to focus on distribution of health burden in the historic time period. And this is a kind of a interdisciplinary study that has partners from state, federal, and local agencies, as well as academia and private sector. It's a very nice group of expertise that we've been able to put together. Okay, now, so for this study, we assessed health risks and health burdens for cardiorespiratory hospitalizations and emergency department visits at the residential zip code level. And then using previously validated PM2.5 exposure data sets, we assess PM attributed health burden and differentiated by impacts of smoke, by age, race, gender, socioeconomic status, as well as annual average background air pollutions at the zip code level. Why do we use health burden rates? It allows us to have like a single metric that encompasses three different things. One is PM exposure, intensity and duration varies over space and time. PM specific risk differences differ by groups, as well as baseline differences in disease incidence between groups. So turning everything into a health burden rate, we can address disparities, compare rates over time, geographies between groups, and so forth. And then we can put it also in the larger context of how does these burdens compare to other outcomes, COVID, cancer, and so forth. Because, you know, on average, most people would not really be able to put in the context what 10 per 100,000 excess deaths or emergency departments mean, unless you compare it to something, anchor it to something that you know, like COVID or cancer. All right, so this slide shows you two maps. One is the distribution of PM on a non-wildfire smoke days, and the other one is the model estimated concentrations of particulate matter on wildfire smoke days. So it's like daily average, right? Well, the one thing that I want to point out here is that there's spatial patterns, and not all geographies are impacted equally. Additionally, you can notice that some geographies are impacted by both wildfire smoke and ambient sources, while others are impacted by one or the other, and only very few areas, these are residential zip code areas, are not impacted by either one of them. Now, there's a smoke, when you look at the frequency of smoke over this period, 2008 and 2016, that I forgot to mention that before, there's areas where we observed 699 days of smoke. That is like a lot of smoke days. But you see that northern part of the state is really impacted by smoke. Now, and there's areas that are impacted by smoke less often, or by lower concentrations. However, they have a lot. These areas often sometimes have a lot of people. So you have to account for person time, right? And then finally, let me see. What I want to say is, in addition to exposure and people numbers varying by geographies, population composition also varies by geographies. This is important because measure of risk, as well as the baseline rates of certain outcomes, like in this case, respiratory and cardiovascular outcomes, vary by composition of population. And I use this particular table we differentiate by main race groups, right? And on average, across the state, we have around 4,000 per 100,000 people, emergency department visits for respiratory and cardiovascular outcomes. Now, compare those numbers in the first red box to the other red boxes. Black population is less than 6%. So if you only accounted for their attributable number, they wouldn't probably, it wouldn't probably be observed as a differentiated impact. However, the baseline burden rate, if you think about only in that group has very large baseline burden rate, 2.5 times the average, right? So the 5% increase in risk in whites and blacks means different number of excess burdens. But if you think about it only in absolute terms, you may not see those numbers, because whites in this case account for 40% of population, whereas black population accounts for less than 6%. Another group that I want to point out to is Asian. Asian population accounts for 13% of population, but only 5% of total respiratory and cardiovascular outcomes. And I want to keep that in mind for the next slide. Now, the previous slide was just the baseline burden rate. But now when we calculate burden rate attributed to PM exposure in the first column, and in the second column to the ambient PM exposure on smoke days, we see that some of those inequalities transfer to the PM burden rate as well. In black population, again, has a very high PM burden rate, which is attributed, which was due largely to that large baseline morbidity. Whereas in Asian population, the burden rate is small, but the fraction of their total respiratory burden that's accounted for by PM exposure is larger, because their relative risks are larger and their exposure tends to be higher because of kind of like the area where they're more populated in. They're found there's a higher percentage of Asian people living near the large urban areas. Finally, I want to show these 4 figures that show that excess ED burden rates for 4 different diseases, 4 different outcomes that we usually study in wildfire smoke studies: asthma, COPD, respiratory, and all-cause cardiovascular outcomes. And what's important to notice here, that there are differences here, but the patterns are largely the same, and that's due, and the numbers reflect those burden rates and the risk differences more than sorry, the numbers reflect differences in risk and differences in baseline burden rates. We can also quantify these burdens over time. If I'm meant to go circle back to the first slide, I showed how PM2.5 has been decreasing over the years, and we see that here in California as well. So naturally, the attributed fraction of respiratory or cardiovascular outcomes follow this pattern, right? This is for asthma and respiratory, CVD and COPD. Now, so it's a nice steady decline. However, if you look at what fraction of that PM burden is observed on days and areas that are impacted by wildfire smoke, that attributable fraction varies much more over time. And in some years, when smoke season is high, then up to 15% of the total PM2.5 burden is could be attributed to wildfire smoke. Now, so if we look at these two figures, the progress that we are making over time, 15% accounts for about 2 years of progress that we're making due to reduction in PM from all other sources. It's kind of like a big number if you're looking only at the worst case scenario. And so this is really all I wanted to talk to you today about, and to show you how significant the impact of wildfire smoke exposure is on the health burden in California, and how these impacts disproportionately affect demographic groups and regions that are already vulnerable to poor air quality. In studying these health outcomes, we found the largest PM burden to be attributed to respiratory outcomes, accounting for as much as 2% of total respiratory emergency department visits and 0.9% of cardiovascular outcomes. And then to note that as we're decreasing, or as we're improving our air quality and decreasing the PM attributable burden from all other sources, the contribution for wildfires is significant and can account for up to 15% of the total PM2.5 burden, right? And when considering disproportionate impacts among subgroups, very often in many groups, we are seeing the same differences that are accounting for underlying differences in morbidity and exposure being translated into smoke impacts as well. So by age group, differences by age group, by race, by geographies, specifically rural, central, and northern California populations have experienced increased wildfire attributed health burden, but many of these areas have also had traditionally high impacts of ambient air pollution. And then that's all I was thinking I would have time to talk about today, but then talk a little bit more about those future impacts for prescribed burning and quantifying trade-off when we see each other again in Chicago.
Terrific. Thank you, Dr. Rappold, for that great presentation. Reminder, if you have any questions, please use the Q&A function or the chat function, although it seems like most of the questions are in Q&A. Anna, you're welcome to respond to some of them if you'd like, but we don't have time for questions right now. We want to go to our next presenter, so I'm happy to welcome Dr. Montrose. Luke, go ahead and share your slides and get started.
That look okay?
It looks good.
All right. Well, it is quite the honor to have been invited to speak with you all today, and I will do my best to follow a really wonderful presentation by Anna. And so the first thing I'll do is just let you all know that none of the work that I'm going to present today could have been completed without a huge team. My lab collaborates with a number of different folks across the US, and we attempt to do good work in the area of field science and lab science related to wildfire smoke. And I've listed some of our goals there, but what you'll see today is that I'm going to sort of tell one story from each bucket. We try to develop sustainable relationships with understudied or vulnerable populations to reduce their risk to wildfire smoke. And then also to, in the lab, develop models where we can try to characterize exposure-induced effects. And one of the areas that we're moving into, which I'll talk about today, is extrapulmonary effects, including the brain and reproductive system. I think we had a really good introduction to why we should be concerned with wildfire activity and the amount of acres that are being burned throughout the United States. We've also, I think, heard a little bit about the contributing factors, such as increases in temperature and decreases in precipitation. And this is an idea that's acknowledged by the U.S. Forest Service and their recent document called Confronting the Wildfire Crisis, where they specifically address the amount of fuel load, which has been increasing, juxtaposed with these changes in temperature and precipitation. And that's what brings us to today's challenge. Anna also mentioned this. I'll just reiterate that while urban particulate exposures are improving primarily in the eastern part of the United States, we see that from this chart from McClure et al., that the Rocky Mountain West and the Pacific West are getting hammered with wildfire smoke, and so kind of going in the wrong direction here, while the rest of the United States is improving. So what's in wildfire smoke, and why are we so worried about this particular toxicant? There's hundreds of chemical constituents that make up wildfire smoke, and I'm going to focus on discussing PM 2.5. Sorry about the automatic transition there. PM 2.5, 2.5 micrometers in aerodynamic diameter, describes a particle that can get deep into the lungs, shown here, where we see that the particles are reaching into the alveolar space. And the EPA recognizes that this is an issue, that this is a challenge, a toxicant of interest, as they've listed it on their criteria pollutants. There's a benchmark of 35 micrograms per meter cubed that is important for an outdoor standard. And you may not have heard of that number before, but you can see that that number exists here in the AQI, the Air Quality Index, and that's the transition between where we go from moderate to unhealthy for sensitive groups. And I mentioned that there's hundreds of different constituents that make up smoke, and those constituents can change based on the burning conditions. And what I mean by that is, what are we burning, and what's the temperature, and what are the other conditions like meteorologic conditions around that burn? These can all contribute to a very complex profile of chemical constituents. This can be kind of challenging to study, and we've developed a lot of models in the lab to be able to more finitely tweak these different conditions and these different constituents. So wildfire smoke has been linked, as Anna pointed out, to both acute and chronic conditions, so I won't spend too much time here. But I do want to point out that among those in our community, those folks who are most vulnerable to this are often those who are at the ends of the age spectrum, so the very young or the very old, those with comorbidities like asthma or COPD. And then I'll also point out, we oftentimes forget about those folks who can't get away from the smoke, those people who work in the outdoor environment. And I'm going to talk about one specific group today. I'm at this Colorado State University, so I would be remiss if I didn't point out also that this is not just a problem for humans, but it's also a growing problem for animals. So in the introduction that I was given, it was mentioned that I do my work in a very wide lens from cell to society. We study biomarkers and mechanisms of smoke, indoor and indoor air quality assessment and communication, and then also data dissemination and creative advancement. And I'm going to touch on each of these today, if not briefly for some of these. I'm going to try to tell two stories, although with limited time, I may have to skim the second story, but I do want to get to questions, so you'll forgive me if I do a bit of skimming. Two main stories here. One is, and the first one is protecting firefighter health and well-being. So if you're not familiar with wildland firefighters, these are a very diverse group of folks and wildland firefighter as a term is very inclusive. You can see that these are all wildland firefighters, but they're all completing a different type of job, and their job actually is predictive of their exposure. I also want to juxtapose structure firefighters with wildland firefighters and point out that their exposure profiles can be different, primarily because of the personal protective equipment that a structure firefighter wears. Although he or she may be in an environment where there may be smoke and other types of chemicals, that breathing apparatus changes it reduces the dose as it compared to the exposure. Whereas for the wildland firefighter, they typically are only wearing that face shroud, which does more to protect them from the heat than it does from the actual smoke. So what do we know about wildland firefighter health as it relates to their job and to their smoke exposure? There's a number of different review studies that have been conducted looking at this question, and we see that there's cross-shift and cross-season changes in lung function that were associated with acute systemic inflammation. They seem to have a greater risk of cardiovascular effects and also increased risk for lung cancer and cardiovascular mortality. Now, I want to try to convince you that there's some motivation to move from the traditional target tissues of the lung and heart and begin looking outside of the lung and heart. And this is because of that systemic inflammation point that I just made. There are a number of different cytokines and chemokines which can be measured in the blood after a wildland firefighter has been exposed to smoke. And I would propose that this is evidence of inflammatory spillover, or in other words, inflammatory factors gaining access to the bloodstream and therefore pretty much any tissue in the body. So what other tissues may be targets? Here's some evidence that I believe suggests that there's that it's plausible that reproductive health could be impacted. From the urban particulate literature, we have a number of different studies that suggest in Chinese cohorts, in U.S. cohorts, and in animal cohorts that there are decreases in sperm following acute exposure to urban particulate. There was a recent wildfire smoke exposure that was done by Rubin et al. In the Portland, Oregon area that looked at repeat sperm donors and found that if they looked following a wildfire smoke event, that those repeat sperm donors produced lower quality sperm. And then from the structure firefighter literature, we have a couple different studies that suggest that there's infertility related to being a structure firefighter, not specifically smoke, but obviously smoke is one of the exposure factors that they're challenged with. So what we wanted to do in my lab was to try to come up with a model of wildland firefighter reproductive health using a mouse. And one of the sensitive biomarkers that we thought we might be able to use was DNA methylation. And so essentially what I'm suggesting here is that the spillover effect may also be seen in mice. And we actually know that from the literature that's been observed. And then what I wanted to do was to see if it was hitting the reproductive organs of the male mice using a sensitive biomarker. And that sensitive biomarker is DNA methylation, which I mentioned. If you're not familiar with this, this is a a methyl group that's added to the fifth cytosine on a fifth carbon of a cytosine residue. And, canonically, what we think of is when we activate DNA methylation, or we lay down DNA methylation, we get a condensing of the DNA strand. And this can be a good thing if this is something that you're trying to do, for example, to try to package DNA in the cell. That's a biologically necessary process. But if it happens in the wrong place at the wrong time, this can be problematic. So the system that we used was at Northeastern University, where we were able to expose mice in a whole body chamber to Douglas fir needles, representative of a simulated wildfire event. We did this at 20 milligrams per meter cubed, which is a pretty apocalyptic dose. And we did a calculation to try to figure out how much deposition we needed to get to reflect 10 years of wildland firefighter service based on a lung volume comparison between a human and a mouse. And we found out that we needed to expose them for about 40 days at 20 milligrams per meter cubed. And that's what we did here. And this is at a smoldering temperature, which I told you earlier is important for the chemical profile. So what we found in an initial sort of survey of DNA methylation across the entire epigenome was that there was more methylation among wildfire smoke exposed mice in their sperm as compared to the fresh air exposed. Now, like I said, this is a sort of a shotgun approach. We don't know which genes this is occurring in. We're doing a survey of the entire epigenome. So the next thing we wanted to do was to try to zoom in a little bit more and use a technique called reduced representation by sulfite sequencing. This is a technique that is enhanced for the types of areas that we would anticipate being functionally relevant for gene transcription, or in other words, gene or protein, the amount of protein that's produced. And so this uses a digestion enzyme that seeks out CCGGs, which is more common in the promoter region of genes. So what we found was, and this may not be initially apparent, but I think once I explain this, you'll see that there is quite an interesting change here. On the right-hand side are the fresh air mice, and what you're looking at is a violin plot of methylation patterns across these enhanced regions, these promoter regions primarily. On the y-axis is DNA methylation with 100% methylation at the top and 0% methylation at the bottom. Those that are 0% methylated, we think of those as being, you can think of them as being genes that are turned on, and the ones at the top being genes that are turned off. And if you look, the wildfire smoke bimodal distribution is actually compressed more towards the center. And this is reflected in the numbers. When we quantify this at the bottom, there were 2,650 regions that were hypermethylated, and that's the shift from the lower portion getting shifted up, whereas 703 or 21% are being hypomethylated, shifted down. So very, very interesting, still not showing us specific genes yet, but giving us an indication that this wildfire smoke exposure is having an impact on DNA methylation. Now, I want to stop here and give you some considerations to think about when we try to make a transition from thinking about reproductive health to offspring health. So would these characteristics that the sperm are now carrying following smoke exposure be conferred to the offspring? And I would suggest that this is that epigenetic inheritance across organisms is plausible but contested, and I'm going to show you why that is. Essentially, the epigenome as we know it gets erased once that sperm meets up with a oocyte to become a zygote, and that's because for that zygote to go on to create billions of new cells, heart cells, lung cells, skin cells, it has to forget everything it knew about itself as a sperm cell. And so you see that here in this grand erasure that happens once the sperm meets up here with the zygote. Now, we do hypothesize that there are a number of genes that do escape this. And so these dotted lines here represent those genes that might escape. Those genes are primarily going to be imprinted genes. And so we also wanted to look to see how many of our 3,000 genes that were differentially methylated were in an imprinted region. And we used the imprint gene database that's curated by Dr. Randy Jurtle. And what we found was that there were 20 genes that were hypermethylated that were imprinted genes. This one gene is actually also counted in this 20, but in that region, it had one site that was hypermethylated and one site that was hypomethylated. So that's why that one has the plus there next to it. But at any rate, we did show that some of our genes that were differentially methylated following wildfire smoke exposure did fall into an imprinted gene category, and thus there is some plausibility that differential patterns could be conferred to the offspring, and this is work that we plan to do into the future to try to confirm that. So as we were producing this data, some interesting papers started coming out on ambient particulate exposure and changes in sperm quality. And this group was able to connect this back to the HPG axis, or the hypothalamus pituitary gonadal axis, shown here on the right, where essentially you get a signal from the brain that is actually telling the gonads to produce or to begin the spermatogenesis cycle. And so what they were able to show was that that particulate from an urban area modified this cascade of events, and they were actually able to identify that a specific transcription factor receptor site for NF-kappa B in the brain was one of the primary drivers of this. So this was very interesting to us. So we started thinking about whether or not this could have any type of impact on what we've been seeing with wildfire smoke and sperm. So we used a different exposure system with a colleague who gave us more ability to collect more tissues. We used a slightly different, this was not an occupationally relevant exposure, rather it was a acute high dose exposure paradigm, 5 milligrams per meter cubed for 2 hours a day for 7 days. And we harvested these animals directly after that 7th day of exposure. Again, this is a whole body simulated wildfire smoke exposure paradigm. And so we wanted to start poking at this cascade of events and see what we could elucidate with this model, trying to understand whether or not the HPG axis was involved in spermatogenesis for these mice exposed to smoke. So one of the first things we said was, well, can we determine whether or not we're getting systemic inflammation? And the first thing we were able to show is that with these mice that serum C-reactive protein was elevated in the wildfire smoke exposed mice. The next thing we were able to show was that this is somewhat interesting. We're not exactly sure why this would be the case, but we were able to show there was morphological changes in the testes, with the testes being larger in the wildfire smoke group as compared to the fresh air group. And we don't know if this is due just to inflammation, or if this is perhaps due to an increase in leakiness of the blood testes barrier and the allowance of fluid to build up in the testes. We then looked to see if we could see hormone levels shifting in this cascade. And we looked at testosterone in the serum and also testosterone in the hypothalamus. Again, this is 24 hours after the exposure, and we did not see an indication that those hormones were changed, but we'd love to go back and do this at different time scales to understand if perhaps we either missed a window or didn't let the exposure go on, we should have let them age a little bit longer, and then we might have seen this build up. We're unsure if this is just is it an artifact that we just didn't see? One thing that was very interesting to us was that when we looked at TNF alpha and NF kappa B, two important pro-inflammatory cytokines in the brain, that we were able to detect an elevation in the wildfire smoke exposed group. So, and this is actually right about the time that we were doing this work. Matt Campen's lab at the University of New Mexico put out something very, very similar to this. So we have pretty good confidence that what we're seeing is an actual exposure-induced effect here, where we're seeing pro-inflammatory cytokines in the brain being elevated. So we're still poking at this story, and we plan to do some more work, like I said, with different timelines into the future, but some preliminary data that suggests that wildfire smoke may be having an impact on both brain and via the brain, perhaps reproductive outcomes. I just want to point out that we're planning on building an exposure system here at Colorado State University. So in the future, we'd love to collaborate on projects in vitro, in vivo, instrumentation, exposure assessments, any of these things. If they're of interest to you, please reach out. Another thing I wanted to point out is that we're going to try to translate these mouse findings. Again, the end goal is to translate this to a human exposure. And so we want to understand whether or not these impacts are observable in a wildland firefighter community. And so we're going to be using an at-home sperm analysis kit to start looking at this story. I also have an upcoming postdoc opportunity that I would just point out if you're interested in supporting the prioritization and coordination of research around wildland firefighter health and well-being. Look out for this in the fall time of this year. I hope to have that out by then. I did say I was going to skim this last story if time was an issue. So I'm just going to give you a brief sneak peek in this, and please reach out if you'd like to learn more about this story, or we can talk about it in the Q&A session. But this is a story looking at skilled nursing facilities. This is a place where folks are very vulnerable. I had a very awesome undergraduate student who wanted to ask the question, are there areas in the Mountain West that aren't getting good real-time air quality data from our regulatory monitoring systems? So I asked her if, with her ArcGIS background, could she overlay maps of air quality monitors, regulatory air quality monitors with skilled nursing facility locations. And this is what she came up with. We found that there were the white circles are a 30-mile buffer around a monitoring site, and anything outside of that 30-mile buffer is the black region, what we deemed a monitored desert based on some EPA guidance. And any red flag is a skilled nursing facility in a monitoring desert. Out of the 777 skilled nursing facilities recognized by Medicare and Medicaid in the Mountain West region, we found that 125 or 16% of those skilled nursing facilities were in a monitoring desert. So there's some opportunities here to provide some better real-time data to skilled nursing facilities in the West. We then launched a project looking at putting low-cost monitors inside of these facilities. And I'm just going to kind of scream through this. We looked at 4 different facilities across an entire year. This is in 2020 from January to January. And what we found was that for certain facilities, these facilities happen to be right next to each other. The blue is the outdoor air quality. The red is the indoor air quality. You can see that there's a big separation with facility 1, suggesting that that facility is doing something to improve the indoor air quality when there's an outdoor wildfire smoke event. You can also see that the wildfire smoke event here dominates any type of particulate in the air throughout the entire year. This August to October season is where we see the spikes. And the facility right down the street from facility 1, facility 2, has a much smaller gap, meaning it's not as good to be inside. In some cases, it was the same, air quality was the same outside and inside. And so one of the things that we want to ask is, what predicts the variability here? Could it be that the building was older, or that the HVAC system was older, or that there's more square footage or penetration points? And in this case, facility 1, which did worse than facility 2, is actually newer and smaller. So it kind of went against our initial predictions, and we need to sort of dig into this a little bit more. We hope to scale this project up and be able to better answer this question of what predicts variability in indoor infiltration. That should say infiltration, not inflammation. Sorry about that. I've identified that building managers are a good entry point to try to improve indoor air quality for all of these reasons. This is within their job duties, and they get almost all, according to some of our survey data, they get almost all of their training on the job. So these folks could be a good source for education and outreach to try to improve indoor air quality. And that's what we hope to do in the future, moving towards a comprehensive smoke management plan, including both training for those building managers, as well as providing more on-site hyperlocal air quality monitoring dashboards. So I want to end with, in case you're thinking to yourself, what is there anything that we can do to protect ourselves from this smoke? I just want to end with, there are lots of things that you as an individual can do. You can avoid activity outside when there's a wildfire smoke event. You can clean the indoor airspace with something like a Corsi box shown here. And you can also try to avoid producing your own indoor sources of air pollution. You can also look at these wonderful web pages to try to stay informed, things like Air Now, or if your state has its own smoke blog, like in Idaho, they have the Idaho smoke blog. You can also research low-cost monitoring web pages. You can buy, if you have the means, you can buy these very cheap and inexpensive air quality monitoring systems that link right up to your phone, so you can be your own citizen scientist. And the last thing I'm going to say is that 4 years ago, I started the Wildfire Smoke Symposium. This year's Smoke Symposium is going to be in Colorado at CU Anschutz. We'd love to have you. We're teaming up with the Mountain West Society of Toxicology. This is going to be a wonderful event themed this year on translational toxicology around wildfire smoke. And with that, I'm going to stop right there. I hope I left enough time for questions. Thank you.
Great, thank you, Luke. Very interesting. And thank you, Anna, as well, for these presentations. We don't have a lot of time for questions, but maybe we can get to one or two questions that we can't get to today. We'll collect those questions and distribute them to our presenters after this is done. But Anna, there were several questions broadly related to exposure and how exposures were characterized in your studies. Do you have any reactions to one or more of those?
Sure. I'm sorry, I didn't see the question specifically, but I can say we use a variety of exposure. The results I was showing were using what we refer to as Harvard PM model, right? D et al. Because there's a lot that we have to know before we can really have really good exposures from wildfire smoke, right? And validated exposure. But we thought that using this metric was something that would increase ability to reproduce results and to compare results across the studies, because it is available to research community. And then we use model outputs only to kind of create strata of smoke, right? Like, low smoke, high smoke, and so forth. So we're not using that exposure to calculate excess burden. We use the epidemiologic analysis based on D et al. in this work. But then there's also a lot of opportunities for like statistical modeling of model calibration and improving the predictions of smoke using models as, just to enhance the predictions. So,
okay, great, thank you.
Little chunks, little pieces of progress, Yeah.
And maybe we have time for one more. I know we're at the hour, but Luke, I assume you can see the two questions that were posted that are related to your study. Do you have any comments now on these?
The two I think I saw, one was on whether or not indoor air quality spikes contributed to the overall exposure paradigm. And I think the most telling way that I can answer that is to look at the entire year. We don't see so red was indoor and blue was outdoor. We don't see spikes of red that surpass blue. So the blue is always going up and then the red is following it. And that, I think, gives us a really good indication that the outdoor air quality is driving the indoor air quality. And if you look up our paper in indoor air, you'll actually see that we did an infiltration efficiency calculation, which actually does, as the questioner suggested, it actually does remove indoor air quality with a calculation that's a little above my head. Of course, I had help doing that calculation with a bioinformatician. So that's definitely something to keep in mind. And I think the other question was related to whether or not the mouse sperm studies can provide any window into our understanding of why U.S. men's fertility has been decreasing over the last 50 years. I think that is that particulate matter certainly could be contributing to the number of factors that can cause decrement in sperm. And so that's something we definitely want to look at. And wildland firefighters may be the canary in the coal mine that help us to understand that as we move forward. So thank you for that question.
I agree. Okay, well, I'm going to wrap up with a few concluding remarks and then I'll let you all go. So thank you for participating in this webinar. In the next day or so, you'll receive an email from ISES that includes a link to an evaluation, and please take a minute to provide your feedback. ISES is proud to offer these complimentary webinars every month. By joining ISES, you'll be afforded other member benefits as well. The next webinar is in May. We'll have registration and details coming soon. You can visit the ISES website under the calendar of events menu for more information and registration details. Also, don't forget to mark your calendar for the 2023 annual meeting in Chicago, which is August 27th through 31st. Abstract submissions are open until tomorrow. So one last time, thank you ISEE, thank you ISES for organizing this. Thank you Luke and Anna for presenting today, and thank you all for joining.
Thank you.
Take care.